we're going to explain the role of ADH. And ADH, or antidiuretic hormone, is a peptide hormone. So it's going to bind to receptors on the cell surface membrane. It's produced in the hypothalamus. So we can see the hypothalamus is going to release ADH. And just as a quick recap, it was pardon, the posterior pituitary. So it's produced in the hypothalamus, but that's by neurosecretory cells. Produced by neurosecretory cells, which then store it in the posterior pituitary, releasing ADH into the blood. That other O was for oxytocin. Okay, so it gets released into the blood and then it's going to travel down. So for every part that we've looked at so far, we haven't actually mentioned any control, really, other than all these processes are happening. We're reabsorbing water, we're filtering the blood, we're excreting urea. But we haven't mentioned any control by the brain. So this is where the brain comes in. The brain, the hypothalamus, it's got receptors, and those receptors are allowing it to work out the osmolarity of the blood. So it's detecting essentially how much water there is versus the solutes. And if we get blood that's too dilute or too concentrated, and by too concentrated we mean solute concentration, so salts and sugar, if there's too much salt and sugar, then the brain needs to switch on and do something. So what it detects is too concentrated, and that leads to the release of ADH, because a diuretic is make you urinate, make you pee. So this is anti-make you pee. So this hormone is going to reduce, you're still going to urinate, but you're just going to reduce water content. Of the urine. So ADH is acting on, it does act partly on this part here, the last part of the distal convoluted tubule, the DCT, but mainly it acts on the collecting duct. Now you can see that the collecting duct is also going down deeper into the medulla. And deep in the medulla, we said there was lots of salt. So because this part here is very salty, especially towards the bottom, if water can move, water will move. So the default position is that water will get absorbed out of the collecting duct. Now, if we absorb lots of water, that would mean we get, thinking about our boxes at the end, we would get less water there. So I've drawn the boxes. If you are dehydrated, then as the filtrate comes through, you want to absorb water out, leaving less water, and what would be remaining would be urea and salt. So we'd get a concentrated urine with more salt per volume and more urea, and this is what leads to the darker colour. So how does that happen then? How do we, if we're dehydrated, how do we get more water? Well, we said the default position is that we would absorb water if we could. So why is water not always being absorbed very much? And it's because the collecting duct is not very permeable to water. So let's have a look at this situation here. Over here we've got the blood, where we're going to absorb back the water. So let's have a look at the two situations. In the top situation, we have less water being reabsorbed. So why is that? Well, we can see here that in the membrane, we've got some proteins, and these are aquaporins. Water doesn't move very well from, this is the collecting duct. It's not going to move through the cells which line the collecting duct. So these are the cells, the epithelial cells. It's not going to move very well through there without aquaporins. So some will diffuse, but not very much. So we need aquaporins, which are, let's just draw a little picture of it from that. They are channels for facilitated diffusion. So if we've got channels, if we put a channel into the membrane, well, here we can see not many channels. That means not much water can be reabsorbed. And that means that we're going to get a dilute urine. So down here is the situation when ADH is released. When ADH is released, it travels in the blood and it goes and it sends a signal. It's a peptide hormone. It binds to receptors, and the signal to this cell is to move more aquaporins to the membrane. And when it moves more aquaporins to the membrane, now we have more channels, and now we can absorb more water. So we get more water being absorbed, and if we get more water being absorbed, then there's less water, and we get a concentrated urine. Okay, let's just have a look at the top situation. So if you're well hydrated, you've drunk a lot of water, then the hypothalamus will not release ADH. So there's no ADH release. And then if there's no ADH release, then these aquaporins, they don't have a very long lifespan, and they will be recycled. So we have a decrease in the aquaporins, and that means water can't move as much, and then the water sticks in the collecting duct, and then we get a dilute urine. So the two other words that we could use, dilute, we could say it's a hypotonic. So there's more water in this solution than in blood cells, whereas when we had ADH, because we've reabsorbed lots of water, we would then get a hypertonic urine. So this is also working via negative feedback, because when we are dehydrated, we get an action via the hypothalamus which reverses the change in direction of the system. We get a return to normal levels. So the final little recap with the essay, we can see that ADH plays a role in osmoregulation, regulating the blood solute concentration. Acting on the collecting duct, it also acts in the late distal convoluted tubule. The hypothalamus, the brain, was detecting the solute concentration. And if the blood was too concentrated, we would then get the release of ADH from the posterior pituitary. ADH would stimulate insertion of aquaporins. The water would then move through, and yeah, good to mention, which we didn't in the last part, it's via osmosis, moves via osmosis. Then we get concentrated urine until, and this is an example of negative feedback, until then the blood becomes hypotonic, in which case it would then, we'd have no ADH released, so water doesn't get absorbed, it's not reabsorbed from the collecting duct, and then we get a more dilute, a hypertonic urine. Okay, that's it.